Hello, good morning everyone. Hope you are doing well. Today's topic is recombinant DNA technology. Recombinant DNA technology is the process of cutting and combining DNA that contains genetic information from two different species of organisms. For the process of recombinant DNA technology, we require different tools. First one is restriction enzymes, and enzymes that recognize a specific short DNA sequence and cuts DNA internally on both the strands. The second tool that we require is DNA ligase, an enzyme that joins DNA molecules. The third tool we require for recombinant DNA technology is plasmid. Plasmid is an extra chromosomal circular DNA molecule found in bacteria, which contains antibiotic resistance gene and can contain gene from foreign organism. There is another process that is bacterial transformation, which is the process in which bacteria takes up and express DNA from outside of the cell. Here is the procedure of bacterial transformation. The first step is mix E. coli cells in calcium chloride solution, which is cold, forms spores on the cell of bacteria. The next step is addition of plasmid. The third step is heat shock treatment. Heat shock is a short sudden exposure to heat to induce intake of DNA which is present in the environment. The last step of transformation is allow E. coli cells to grow on the media. Here is the diagrammatical representation of ampicillin containing media. Here is two bacterial cell. One cell is transformed with plasmid, or another cell is not transformed with, which grows on the media overnight. The cells that transform with the plasmid are grow on the media, or the another one cell that is not transformed with the plasmid are unable to grow on the ampicillin media. The first step of bacterial transformation is the ampicillin sensitive E. coli cells in lag phase of growth are transferred to cold calcium chloride solution. The second solution is ampicillin resistant plasmids are added to experimental cells only. The third step is cells are heat shock at 42 degrees Celsius. Some of the competent cells take up the ampicillin resistant plasmid and are transformed. The fourth step is to treat the cells are spread on the agar plate containing ampicillin media. Ampicillin in the agar media kills the bacteria that lack ampicillin resistance genes. The fifth step is to incubate the plates for 24 hours, and last is only colonies of E. coli that have been transformed with the ampicillin resistant gene will able to grow on the media. There is another one technique that is DNA profiling, also called as DNA fingerprinting. It is the analysis of fragment prepared from the DNA of an individual, which can then be used to distinguish from the two individuals or between two individuals. Here is DNA profiling can be done by using PCR technique. PCR is a polymerase chain reaction which is invented by Carey Mullis. Is the analysis of DNA sample by making many copies of specific DNA sequence. Here are the steps of PCR. First step is denaturation, second is annealing, and third is elongation. For the multiple copies of DNA, we have to repeat three steps of PCR. In first step, we perform a denaturation of template DNA. Here you can see that there is a double stranded DNA which converted into a single stranded with the help of heat at 95 degrees Celsius. Here are two strands are formed, that is first is 3' to 5' and another one is 5' to 3'. The second step is annealing of primer. Primer is adhered at both the ends of DNA at 3'. The third step of PCR is to incubate DNA polymerase and nucleotides. Incubate DNA template with the DNA polymerase and nucleotide. And for the multiple copies of DNA, we have to repeat all the three steps. Here is the graphical representation of the PCR. In first step, you can see that there is a template DNA. In first cycle, the template DNA or double stranded DNA gets separated into single stranded, and each strand is used as a template or a mother strand. For the multiple copies, we have to perform various cycles. Here is a PCR temperature cycling. In the first step of PCR, that is denaturation, we require high temperature is about 95 degrees Celsius, and for the second step we have to reduce the temperature up to 55 degrees Celsius. For the third step, that is extension of DNA strands, we have to increase the temperature up to 70 to 72 degrees Celsius. Here are some component which requires for the PCR. First one is DNA polymerase, enzyme involving replication, which is Taq polymerase, extracted from the Thermus aquaticus. The second is target DNA, which is DNA molecule to be replicated. The third one is primer, which is short single-stranded DNA sequence complementary to the ends of the region to be amplified. The last one is and the most important one is nucleotide, which bases incorporated into a new strands of DNA, that is adenine, thymine, cytosine, and guanine. PCR can be used in cloning, sequencing, forensic, and diagnosis of disease and infection. Here is another one technique, that is electrophoresis. Electrophoresis is the technique that uses as an electric current to separate molecule based on charge, size, or shape. In electrophoresis technique, we use agarose gel, that is semi-solid matrix through which molecule migrate. In electrophoresis we use agarose, which is polysaccharide derived from the red algae. In this diagram, here is diagrammatic representation of agarose gel electrophoresis. Firstly, we have to cut the DNA into the DNA fragments, so then allow it to migrate from a negative charge to positive charge by means of electric current. For the visualization of DNA that run on the agarose gel can be done by using ethidium bromide, which is intercalating agents or intercalate between the DNA bases, which shows a fluorescence in the presence of UV light. There is another one dye, that is EZ vision dye. In this last slide, we can see that there is a diagram representing gel electrophoresis. Here is the DNA fragment that are run on the different positions. We can see that there is different position. There is the molecule which having a high molecular weight are near to the wall, and the molecule with low molecular weight are moved toward the positive end. Thank you.